Our next presentation is by Alex Roman-Cabrera. I hope I pronounced that correctly. Hi, Alex. So Alex performed his bachelor's in psychology at the University of Lleida in Spain, and his master's in neuroscience at the University of Barcelona. He is currently a PhD student at Tel Aviv University under the supervision of professors Anat Baranea from the Open University of Israel, Inon Shalf, and Abraham Chefetz from Tel Aviv University. Alex is doing his PhD within the Chicken Stress European Training Network, part of the European Union's Horizon 2020 Research and Innovation Program. The objective of this network is to assist egg producers to attain the highest possible welfare for chickens. And as part of this project, Alex is studying the effects of various conditions, such as age, social group size, and domestication on brain plasticity in chickens, which has been suggested as a potential welfare marker. Really interesting research. Alex, you can share your screen. Great. Alex, if you just need to unmute, we can't hear you.
Okay, can you hear me now? Okay. So I'm Alexander Roman-Cabrera, and I'm going to talk about the brain plasticity of the chickens, the adult brain plasticity, as a potential measure and biomarker of stress. So, as you probably already know, the welfare in poultry industry is something that is under debate currently, and there's a lot of debate about housing conditions, if it's better cage systems, free-range kitchen systems, the transport of the chickens, and different health like kill ball damage or mites. That's really common. There is a lot of different debates, and to try to assess this debate, to try to reach a conclusion, we need to be able to determine the stress of the chickens. And for that, we need a reliable stress biomarker. There is different ways to measure stress, but we need to take into account that chickens as prey animals are animals that tend to hide their stress because it shows vulnerability, and if usually in a natural environment, this will mean that they are harmed preferentially. So it's really difficult to check their stress by their behavior, although possible. But a reliable biomarker, something that we can just directly test in chickens, it will be really useful. There is even the stress biomarkers, and they can be divided more or less in acute and chronic stress ones. Acute stress, like that you are able to measure the stress of an animal for a period of some seconds or even days. We have like blood samples, corticosteroids measured in blood, in the eggshells, in the feces of the animals, but there are different methods. However, chronic stresses are a little bit more complicated. Although there is techniques like further corticosteroid measurements that allow you to measure the stress of the chickens that has happened for weeks. It's really difficult to measure for months. So how can we test if a chicken has been stressed for like months or even all their lifespan? Idea that has been proposed is adult brain plasticity, since it's a biological process that happens across the age of across all life of chickens, and it takes a long time to change. So what is adult brain plasticity? Adult brain plasticity is the capability of the brain to change their structure and function in order to adapt to the environmental changes. One of these environmental changes that can be is stress. So adult brain plasticity has been related to animal welfare and stress, but also with behavior and communication. For example, different members in different hierarchies in the pecking order can, or it has been suggested to have different brain plasticity, learning and memory, of course, and brain regeneration. We are going to focus in animal welfare and stress. Adult plasticity has a lot of different processes, but we are going to talk about neuronal replacement. Neuronal replacement has 3 different processes inside. The first one is cell proliferation. So if we have if we look at the brain of a chicken and we cut the brain, we're just calling a coronal section, so in this direction, we will see the ventricular zone. That is the zone where it's like the factory of new cells. In this area, the new cells are born. You can see here that they are labeled in black. These are new cells, cells that have been born in the last few hours. After these cells, in a process called neuronal recruitment, travel from the from these factories to all the brain and different brain regions. This is called the neuronal recruitment. After some of these cells die, letting space for new cells to come. This is the apoptosis of cell death. This is a really balanced process in which more or less they burn the cells that die and helps the brain to destroy cells whose connections are not efficient and substitute them with cells that can grow new connections and that allows them to adapt to their new environments. Cell proliferation and cell apoptosis, cell death, has been already related, although more studies are required, to stress. However, there is a debate about neuronal recruitment and especially which technique to use to assess neuronal recruitment in chickens. This is something that we will try to discuss in this research. So we want to focus on 3 different brain areas. They are called the median stria 2, that it's a sensorial and motor area. Everything that is related to the senses, you know, and see, hear, smell, it's related with the MST, and also motor. Any movement, it's processed by the MST. The hippocampus is a really known area that is related to memory, but also it's a limbic area, it's a structure linked to stress. And the last is the midopallium caudale. It's a really big area that is related to cognition and high cognitive tasks in general. So thinking. We try to make this broad spectrum to have try to represent all different aspects involved in stress and stress behaviors. So, which is our main objective? So it is to study the cell proliferation, neuronal recruitment, and cell death in laying chickens, and study the techniques that we have to check them, to assess them, to be able to detect them, and then establish if they can be used or not for stress. In our study, we are going to bring chickens from a commercial setting, from a commercial producer. We will keep them in our experimental cages for 3 weeks until they reach an acclimation. And after that, we will allow 3 weeks for the neuronal migration. The cells more or less need 3 weeks from the moment of their born to the moment that they are in their final destination. We will collect brains after these 3 weeks for cell proliferation, and after the 6 weeks of the experiment for neuronal recruitment. So we will use different techniques. For cell proliferation, we will use BRBU, bromodeoxyridine. That is it's a technique that has been, it's a gold standard to measure cell proliferation. It's a technique that is really established, and we want to confirm that it can be used in chickens. In neuronal recruitment, there is 2 different techniques that are being using that are used in birds, BRDU and DCX. We will try to assess if there is difference between these 2 techniques and see which one is better. And also cell death, that the gold standard is Cas5. So from our 10 chickens that we will use, 3 of them will be used for cell proliferation, and the other 7 for neuronal recruitment and cell death. We will perform different immunohistochemistries, and BRDU for cell proliferation, since it's the protein that the molecule that we are trying to label in the ventricular zone, that is the factories of the new cells. And for the neuronal recruitment and cell death group, we will performed BRDU, DCX, and case-based immunohistochemistry in the 3 brain areas of the study. Regarding our results, well, as expected, we found cells in the VZ, so BRDU for cell proliferation can be used. As you can see here, these are some new cells that we detect, and we saw some phenomena that are already known, and for example, a decrease in the number of cells per millimeter in the ventricular zone across the rostrocaudal axis, something that has been already reported. So BRDU is a check. But our surprise, or big surprise, comes when we were analyzing BRDU for neural recruitment, since no BRDU cells were labeled in any of the 3 regions of the that we studied. This is completely in contrast, and it's a really surprising finding because it's against what we know of cell proliferation in the ventricular zone. As I told you, the same technique works for cell proliferation in the ventricular zone. It's against anything that is known in the literature of neural recruitment in younger chickens. And it's in fact different from any other vertebrate that has been studied. Although all these results may seem that there is a methodological mistake, that we just make something wrong, and this is why we have this result, we have powerful arguments that say that probably is not a methodological mistake, but a real result. First of all, the BRDU, the chemical that we use for cell proliferation, was the same. So if it worked for the cell proliferation group, it has no sense that it's not working for the neural recruitment group. Second, we use zebra finch control. We performed this experiment with zebra finches, and the experiment shows different results. And after, we saw BRDU positive cells in the studied regions, but they were not neurons. So other cells that there is in the brain, like glia, they were being recruited, but neurons, that is what we were looking for, they were not being recruited in these brain areas. And also, BRDU positive neurons were found. The problem is that this was in other brain regions that we were not studying. This is a surprising finding, especially if we take into account the DCX result. That was the other technique that we were using for exactly the same, in which, as you can see here, this is the black column is the DCX. We saw DCX positive neurons in the brain areas studied. But I also really interested in the comparison between the DCX and the caspase-3. Caspase-3 is the cell that marker, and as you can see here, there is more cells that are being recruited than cells that are dying. This is a really surprising finding too, since there is the assumption, the breed dogma in birth at least, that there is a balance between the recruitment and the apoptosis. And here we can see that there is a statistically difference in the cells that are being born and the ones that die. Or a hypothesis that maybe that the comparison between the techniques is not appropriate, since maybe DCX is expressed longer than caspase-3. DCX can be expressed for weeks and caspase-3 for just a few days. So maybe this is the answer to this particular question. But let's go to the main discussion, that is why there is a difference between these 2 techniques. This brings us 2 different questions, but both really important. Why there is no BRDU neuron? So why the cells are being born but are not being recruited if there is cell proliferation in the ventricular zone? And why there is DCX neurons, that DCX is a marker of migration. It's an indirect marker of cell proliferation, it's a marker of migration. So while the cells are being born, they are traveling to places, but they are not arriving, at least not arriving alive to the final destinations. The first question, why the cells are being born and but after we don't see them 3 weeks later? It might be a domestication process, that during the domestication we have go so far in the selection of the traits that we design in the chickens, that of course the traits that we design is more productivity or even more health, but it's not in any case that have more memory or more navigation capabilities. I think that for a wild animal is quite important. or it's maybe an effect of the age, since the chickens that we were testing were 80 weeks of age. That is more or less the time that chickens begin to decrease their production. They are beginning to be older. And the second question, why these two techniques are in contradiction? One technique that labels new cells and that are being recruited, and the technique that usually how are migrating the cells are different. This is what we thought. Maybe the CX cells, maybe the answer is that the cells are being born, they are traveling, but they are just dying before arriving to their final destination. So we are seeing just the migrating cells, but we don't see the final destination cells because they are already dead. Or maybe there is other aspects of plasticity that also express the CX. It has been reported that the CX can be expressed in things like dendritic reconfiguration, so other ways of brain plasticity that are not the born of new cells. So just for a summary, BLDO technique that for a long time has been considered the standard in chickens regarding neuronal recruitment may not be applicable in chickens with study stress. But other like DCX seems a potential biomarker. The other one is that brain plasticity might have been altered by domestication, and this is a potential new welfare problem that we need to study. So then the limitations and future research that we were proposing, first of all, the age of our chickens is something that we noticed, and maybe the chickens that we had were too old to really be recruiting cells, although it's known literature that it's a process preserved across their whole lifetime. And also the breed of our chickens that was a lion chicken, that is a highly selected breed of chickens, and maybe a red jungle fowl or a more wild species will not have the same results. We are currently working on both of these problems, and we hope that we will have answers for these mysteries. So thank you very much. Here you have my contact information if somebody has any question. Also, I will answer them with pleasure.
Thank you, Alex. We only just have one question, but at first I'd like to ask, Binti, did you describe the housing conditions of the chickens you examined?
The housing conditions were a cage that we have like 2 per 3 meters, more or less. So they are like, and they are in the zoological garden of Tel Aviv University. So they have like natural life, and the temperature are like common here in Israel, so quite cold.
So there was a special treatment to cause them stress. That's basically what I'm asking.
No, we wanted just to study if the techniques work on chickens. We were more focused on if the technique work on chickens, and after the next step was if they change under stress.
Okay, so someone asked, how do you see this method being implemented as a practical assessment tool for welfare? Now, there's obviously a lot of work to still be done and see if it really.
So if the technique work, and also it's good for, and also it's changed under stress, that it has been tested that yes, some of them change under stress. The advantage of this technique, of course, is the ability to check what happened to the chicken across all their life. It's not like blood that you have information what happened to the chicken 10 minutes before, but if you are stressing the chicken 10 minutes, you will change also the corticosteroids in the blood. This technique allows you to see if the animal has a relatively good welfare across all the life, because changing the brain, at least one of these processes, is really difficult. The bad thing is, of course, that you need to kill the chicken to do it, because you need to assess the brain. So the idea will be that just like some focal animals are sacrificed in a terminal, like in a really big commercial setting, you just kill some of the animals, and then since it's not behavior, the brain tend to follow a lot the normal distribution and these kind of things. It's a really stable. So if 10 chickens in a 2000 cage has like one pattern, probably all the other ones have the same one. So you can just check selected individuals and then generalize these results to all the cage.
I actually have another question I just thought of. Why only laying hens? Why not try looking at that in broilers who live a shorter life?
Mhm. Yeah, this is another step that we are going to implement now.
Oh, you
That,
oh, okay.
that broilers or even other species that like the farm animals that has been like more traditional farming that has been selected less extensively, probably they will have different results.
Well, thank you. It's very, very interesting. Different take on animal welfare assessment. Thank you so much. We'll now take a short break, the last one for the day. Just about 10 minutes. We'll be back here at 3:20, 3:21. We'll see you then. Hi, everyone. I apologize. We're having some technical difficulties, but we'll start now with our next presentation from Enbal, Dr. Enbal Ben-Tal Cohen. She's a senior scientist in the lab of Dr. Yuval Cinnamon from the Agricultural Research Organization, the Burkani Institute, together with NRS Poultry. Enbal is the leading researcher in the project entitled Ending the Practice of Sorting and Culling of Billions-Day-Old Male Chicks, which you will now present. We're very excited to have you, Enbal. Go ahead.
Thank you. Thank you, Liran, for the introduction. It's a pleasure for me to talk in this excellent meeting, and I would like to thank you for giving me the opportunity to talk. The subject of our research is focusing on one of the most devastating animal welfare issues around the world, which is the need to sort and cull billion-day-old male chicks at the poultry industry, at the layer table egg, at the layers industry. So in chickens, there's two kinds of breeds, main breeds, broilers, which are reared for meat, and layers, which lay the table eggs. These two breeds are completely different. While broilers are selected for rapid growth, layers are selected for extreme laying abilities. And together, the two Together, the poultry industry provides over 30% of the global consumption of dietary protein of human consumption. While broilers, both sexes in the broilers are needed for in broilers, both sexes are needed for meat. However, in the layer industry, only female are required, and males, which are very small and unfitted to the meat industry, are a byproduct of this industry and are culled in the days they born. This phenomena is vast. Every year, 7 billion male chicks are culled at the days they hatch, and this is also they cull sometimes in human and waste them by various means. Financially, the industry, this is also very costly. The industry is investing $1 US for each male chick which is culled, adding to $7 billion losses every year. The need for incubation and discarding the male chicks also harm with the environment. Committed to solve this problem, many countries, including the USA and the EU, have a lot of laws to stop this culling. In the 2015, the German government declared a law to prevent stocking male culling until the end of 2016. However, since no practical solution for this problem was proven, the law was cancelled in 2019. The global increase of 50% in demand for in the next 15 years for egg consumption highlights the need to find a solution for this problem. Looking at this number, this is the most devastating animal welfare issue around the world. Throughout the years, a lot of attempt was made to find a solution for this problem. This attempt focused on biasing the ratio between males and females, developing a dual purposes breeds in which females will lay egg and the male will be used for the meat industry, and many other sorting strategies, including discrimination at very early development between males and females. One of the reasons why this discrimination between males and females is very problematic stands on the fact that until very early stages of development, males and females are looking the same. Here you can see, to show this phenomena, here we can see a 3D model created in our lab of a 7.5-day-old male chick, a female embryo. You can see that it is already very developed, containing a brain, eyes, wings, and legs. In chickens, males have two gonads, but females have only one gonad standing on the left side. However, if you look at this embryo, you can see that there is two gonads encircled with red lines. This is a female embryo. So even in this late stage of development, it's not possible to discriminate between males and females, even in this high resolution technology. Despite all of these attempts, today there is no available solution. In the last year, we came up with a solution to tackle the culling of male chicks to prevent them from hatching. Our solution is based on sex determination in chickens, which is determined by the segregation of the sex chromosomes. In chickens, there is one set of sex chromosome in males, which is a Z chromosome, and in females there is two different chromosomes, Z and W. When crossing between females and males, the gender of the embryo will be determined by the mother sex chromosome. If the W chromosome segregates, the embryo will be a female. And if the Z chromosome will segregate, the embryo will be a male. So all the males and only them will have the Z chromosome from the mother. If we introduce a lethality-inducing gene into the Z chromosome, here marked in red, all the male embryo will stop developing, while the female embryo, which gets the unmodified W chromosome from the mother hen, will develop normally and hatch. However, in order to avoid the problems that the mother hen and the generation above her to be sure that they will be alive, we need to control this embryonic lethality gene. This is done by a unique molecular mechanism, which controlled by light. This is the optogenic system. The optogenic system, when eggs will illuminate with the blue light, the embryonic lethality gene will be induced, and male's embryo will not develop. However, the female will get her Z chromosome from her father side and W chromosome from her mother side, and a Y type W chromosome from her mother side, and then she will develop normally, and also will lay a normal eggs, since she is a non-GMO wild type animal. This crucial aspect of non-GMO products is very important in our solutions, and is also recognized by the regulation authorities in Europe. Recently, the European Commission, EFSA, has confirmed that the layer hen and the egg produced by her can be considered as wild type and can be marketed immediately. Also, in the recognition by the Compassion in World Farming, the CIWF, have provided us guides and support. So our solution is benefits from a deep understanding of the industry structure and needs, and have a lot of advantages. Since it's based on genetics, it is expected to be 100% accurate. It does not interfere with hatcheries workflow. It is applicable with different chicken strains and egg types. No time sorting steps of male chicks, and saving 50% of energy and space required for incubation, and no dead male chicks to dispose will benefit with the environment. And the very important aspect is that the product is a non-GMO. And very importantly, the practice of sorting and culling billions of male chicks will end. So in the recent making a transgenic chicken is a very complex task, and is only created in a few centers in the world. In the last 7 years, we were working to create the genetic lines in our lab at the Volcano Center. The heroes of the process is special cells called the primordial germ cells, which are the cells which will form the eggs and sperm of the sexually mature chicken. These cells are purified from the embryo, cultured in vitro, and the genomic modification will be created at the Z chromosome. Then the genomic modification will be validated, and positive colonies will be transplanted back to a surrogate embryo. The embryo will hatch and will form the chimeric chick, which after screening will produce the founders that will introduce the Z chromosome into their genome. So what we inserted into these cells, I will now describe the DNA construct which we introduce into the Z chromosome. This construct is named the Holy Grail construct, and it consists of few elements. Two homology arms, which direct recombination to the Z chromosome, a light-inducible optogenetic system, which induce a lethality-inducing gene. All of this driven by a single promoter. And additionally, we put two regulatory elements called the LOX-P sites. So briefly about this optogenic system. These are consist of two separate proteins, which are activated by light, and each one of them fused to an inactive part of Carrera recombinase enzyme. When induced by light, this optogenic system optogens protein dimerize to form an active CRE enzyme. The CRE enzyme is an enzyme which is responsible for directed genomic changes, and by excision DNA is sequence from the genome between two specific recognition sites. These are the log sites that I showed in the previous slide. So back to our construct. When we activated, when it's activated with blue light, Carrera recombinase enzyme will be activated. This will lead to the excision of DNA inside between the log sites, and resulting in the activation of embryonic lethality inducing gene. So eggs will be set in the incubator, blue light will be activated, and the male embryo will stop developing, while female embryo will develop normally. And this will end the practice of sorting and culling the day-old male chicks. Lastly, I would like to thank all of our lab members, a special team with which are too many to mention, but each one of them have some contribution to our work. And also to Dr. Weyl-Feinstein, which care for all of the team. I would like also to thank Dr. Shelley Druyan and her team for great help and collaboration. This research is supported by the Kidum unit at the Volcani Center and is funded and commercialized by the NRS poultry industry. And lastly, I would like to thank you for listening, and we'll be happy to answer your questions.
Wow, Enbal, I can't tell you how you are here in the animal welfare department. Once this, hits the market, this would solve such a huge problem. That's amazing. Surprisingly, there are no questions about the actual mechanism. I will. Someone did ask, when will this actually be, be usable? When will this be out and used in the open?
So we are working very hard to transform all this genetic modification, which were not so easy. And we already have a lot of proof of concept of our solution. And we are now in stages of commercialization with an NRS company, which funding us.
That's great. So good luck to you and chicks. Hopefully. Thank you so much.
Thank you.
Okay, we'll move on to Aviv Asher. He's our next presenter. Aviv is the head of the Animal Science Unit in Migal Research Institute and the Northern R&D. Dr. Asher is a physiologist, nutritionist, and chronobiologist of ruminants, and much of his scientific research has to do with dealing with factors that affect feed and production efficiency, including the correlation between feed efficiency and the welfare of livestock. During the last 8 years, one of the main issues that Dr. Asher has been investigating is the influence of modern artificial light, mainly LED illumination, on the welfare and production of dairy and beef cattle, as well as sheep and goats. Today, Dr. Asher will share with us some innovative insights regarding the effect of light pollution on the welfare of livestock and their relation to human health. So, Dr. Aviv, stage is yours.
Thank you very much. Just the share screen.
There we go.
All right. So thank you for the introduction and good afternoon. I'm going to speak about the subject of the dark side of the light, or if I can make it more simple, the effect of modern artificial light, especially LED light, on the welfare, production, and feed efficiency of livestock. And first of all, I want to talk a little bit about light and its properties. And light is electromagnetic radiation within a certain portion of the electromagnetic spectrum. And the properties of the visible light is intensity, direction, frequency, wavelength spectrum, and polarization. And we have a visible light. As humans, we can see wavelengths between 400 to 700 nanometers, and livestock can see a little bit narrower than it until 650 nanometers. And our brain interprets the wavelength into colors. For example, short wavelength, the brain interpreted into blue, and long wavelength is interpreted into red, but they can't see the picture or the vision. And one of the very interesting thing that 7 years ago, there was a discovery that we have retinal ganglion cells that are only sensitive to short wavelength, and they're signaling the master clock if we are in dark, in the night, or during the day. And they're not image forwarding like the rods and the cones in our eye. And it is very important to understand because there is a massive impact of this circadian rhythm of night and day, which influence our master clock. And our master clock is delivering the signal by a mediator, which is the melatonin, the hormone melatonin. And melatonin is a super hormone because it's multifunctional neurohormone that is involved in many physiological functions, including regulation of sleep and wakefulness. Also is very powerful antioxidant, one of the most powerful antioxidants in nature. Is also it has a very important part of the immune system and the inflammatory support, and it even can help using treating cancer and even suppress growth of cancer tumors. But something that we need to understand is when a short wavelength illumination is reaching our retina, then melatonin is stopped being produced. Almost is not produced, and we'll see it in 2 or 3 slides. And I'm going to speak about light pollution. It's a big word, light pollution. And light pollution is the negative effect of the modern shortwave illumination. And during 2012, the American Medical Association declared that the environmental factors that cause misalignment of internal physiological rhythms with external environmental conditions are referred as circadian disruptors, or can call or declared as light pollution and as a stress factor. So there is in here we can see that we are, as humans, are very much affected by the new modern illumination because all the screens, the LED technology has the most dominant short wavelength, and it suppresses 80% of the melatonin, and it has a very crucial effect on our health and on animal health. And there are many problems now that there is a massive research regarding to them. And lack of melatonin cause sleep disorders, anxiety, depression, diabetes, and it can increase diabetes, cancer. It elevates the risk of breast cancer. It also can cause cardiovascular diseases, immunological disorders, and also obesity or night eating syndrome, which we are almost all of us aware of it. And so it's a big issue, and there are a lot of experiments regarding to humans. However, what happened or how does it affect the livestock? Because we are forcing them to be exposed to artificial light at night. And we have a very slim information regarding to this point. So I want to show you 3 experiments that we've checked the influence of the modern illumination, especially LED light, LED light. And the first one, we checked it on dairy cows, and we took 90 cows, divided them 3 groups. First group, you can see it in here. The second group was illuminated with red illumination, red LED illumination, and I must mention that the long wavelength, they do not interfere, they do not suppress the melatonin. So that's why we want to check them also. And the control group was cows that were exposed to natural light at night. So regarding to their performance, we can see that milk yield was at the highest level with the cows that were exposed to natural conditions, and compare it to the white lead and the cows that were exposed to white lead, and they had the lowest amount of milk. And we looked at their consumption, and we didn't see any differences in the feed consumption. However, when we calculate the feed efficiency, we did see that the cows, the feed efficiency of cows that were in natural conditions were significantly higher than feed efficiency of cows that were illuminated with white lead. Another thing that we saw is a higher percentage of fats in the milk in the natural conditions compared to the illuminated condition or the white lead condition. And a very important thing is the somatic cells. And we could see that the group of the cows that were illuminated with white lead had much or significantly higher somatic cell count compared to the cows that were exposed to natural light or red light. Another interesting thing is regarding to their heart rate. We can see the heart rate belts that were used days, and we saw something very interesting, that the white lead caused an increase in the heart rate. It also changed the rhythm of the heart rate compared to the heart rate of cows that were in dark or in red light illumination. So we can see an interfere, or we can see there is an effect on natural reconcentration of melatonin. And we saw, and you can see it in here, that the group that were, the cows that were exposed to white lead had the lowest concentration of melatonin in their milk. The melatonin is being secreted in all the body fluid. So we can check it in milk, in plasma, and in urine. Another experiment that I want to show you is regarding to experiment, which is very similar to the cows, and we checked two kinds of elimination. In this case, we checked the white lead and the dark condition, and compare between them. And we did it during the second time, and you can see it in here that when we checked their growth, so we didn't see significant differences between the illuminated calves and the calves that were in darkness. However, we did see that the dry matter intake increased in the case of calves that were exposed to white lead. And like in the cow's case, we saw that the feed efficiency of calves that were in darkness was much higher and significantly higher than the feed efficiency of calves that were exposed to white lead. We didn't see any change or any differences in their digestibility, but when we're looking once again on the heart rate, we can see once again the elevation or the excitation of the heart rate, and we can see a much higher level of the heart rate in the of the calves that were illuminated. So this is the second time that we were able to see it. And another thing that we check is the behavior by taking their steps. And you can see in here during 24 hours the behavior of the calves and their activity. In here, the two peaks are the time that they are receiving milk, so they are very excited. And we can see the major difference is in here during the dim light. And during the dim light, the heart rate of mammals, also us, and the blood pressure is rising and reaching a peak. And so the calves or even people are more active during these hours, and they are reacting to the change of the wavelength. And in here you can see that they did react. The calves that were under dark condition did react, but the calves that were under white lead condition did not react at all. So this is another interference that we could observe. Another thing, we said that melatonin is a super or is very strong antioxidant scavenger, and we can see it also in here in the plasma. And we can see that calves that were exposed to dark condition had much higher oxidant capacity than the one that were exposed to light. Another thing is the energy expenditure. We measure it by measuring heart rate and oxygen consumption. And we can see in here the calves that were under the white light, they had a higher energy expenditure, which can explain why they had overeating. And we did not see it transform into growth. So we can see the energy expenditure, which it was very much affected by the light. The last experiment that I will show you is regarding to goats, dairy goats. They are very sensitive to changes in light. So it was very interesting to check them, and we did the same, like in calves, the same experimental model, and in the white lead group, and also 15 goats in the dark group. And in here, we see a totally different picture than in cattle, or than the reaction to the light. You can see in here that after the lightning, which was occurred in here, in this spot, there was a decrease in the feed consumption, dramatic decrease in the milk yield. They also lost a lot of weight. This is the body weight. And we can see down here the heart rate of only the illuminated goats. And you can see the acceleration of the heart rate after we turn on the lights, during the night, which is very, very interesting. In here, you can see the comparison between the two groups. And you can see in here the differences that of the heart rate, which was much higher, once again, in the illuminated goats. Another thing is we checked the melatonin concentration, and we saw that the concentration of melatonin, which received a peak during the night, was much higher in the plasma, in the plasma of the calves that were exposed to dark, compared to of the goats that were exposed to dark, compared to the goats that were exposed to white lead. So there is a melatonin suppression, and we can also see it in the milk. So this is another thing that surprised us. So all in all, we can see in goats, it's very clear that there is a stress factor. The light is a stress factor, because we can saw that it really disrupts the rhythmicity of the endogenic parameters and behavior. So in summary, we can say that short wavelength illumination at night disrupt internal physiological rhythm of livestock, including disrupting their natural behavior, and has a negative effect, like we saw on the somatic cell counts, on the health, performance, and even feed efficiency. It also elevates or cause an excitation to the heart rate and to the energy expenditure of the animals which are exposed to it. And there is a suppression of melatonin because of this exposure. We found that, and we are still examining it, that long wavelength illumination at night can be a solution, and to the white lead. And if there is a problem to shut down the lights during the night, so it can be a practical solution. And at the end, we can see here a win-win strategy, because when we are preventing the animals the exposure to the white lead or to the modern artificial lighting at night, we're elevating their welfare, but we are also making them more profitable, which is very important to the growers. And we're saving energy if we're not turning on the lights. So it's definitely a win-win. I want to thank all the people and the collaborators that work with us, and still we are working on it. And I want to thank you for your attention.
Thank you, Aviv. Just a couple of questions. Sorry, I lost it for a second, just a sec. Did you measure the impact of the LED light on the total energy use of the animal, not just the production? Was one of the questions.
Yeah, we did. We did, because we measured the heat production of the animal, and we measured their performance, so we can look at all the energy balance components. And that's the reason that I said that we could see that the energy or the overeating, for example, in the calves, were not utilized into the product or into the production pathway, but the maintenance demands, they were elevated. And that's, yeah, we definitely do that. We measure it.
Okay. And did you check for any other behaviors, maybe social behaviors, the effect it has on maybe allo grooming or anything like that?
Not yet. We're going to do that soon. We've checked about, but I didn't have enough time to speak about it, but we checked it on the eating behavior, and we definitely saw an effect on eating behavior that can explain the overeating. Yes, but we're definitely checking Weyl, and we're checking the many more behavioral parameters.
That's great. Thank you. It just occurred to us that in all the legislation, we know they only determine the minimum lux that you can have for an animal and not the maximum. So maybe that will serve for future legislation. Thank you very much.
Thank you.
Our last presenter of the day is Sarah Weyl-Feinstein, who is a veterinary officer in the Veterinary Services of the Western Galilee District in the Israeli Ministry of Agriculture and Rural Development. She is the district referent for animal welfare under the Animal Welfare Act. She earned her DVM degree at the University of Veterinary Medicine in Košice, Slovakia, earned her PhD degree in biology from Haifa University and the Agricultural Research Organization, Vulcani Center. And now she is a resident at the European College of Animal Welfare and Behavioral Medicine. Sarah.
Hi, everybody. Thank you, Liran, for the kind presentation. So I will share the presentation with you. So I am going to present a project we performed 3 years ago, which speaks about the perception of farmers in the dairy farms regarding practical welfare issues. And actually, it's a part of a larger welfare project, which we performed by cooperation with all the professional organizations in the milk industry. So there were representatives from the Ministry of Agriculture, the Veterinary Services, the guiding unit. The next slide, Liran. The second slide. Yeah, the Private Veterinarian Corporation, which is the Haklait, and the Farmer Corporation. Okay, we can continue. The aim of this work was to examine among the Israeli farmers what is their awareness to selected animal issues. Actually, the background for that was the writing of the calf holding regulations, and we needed to create some starting point regarding welfare data in the farms, because we didn't have yet a national database. So in order to measure the improvement we do in that domain, we wanted to create some starting point and then see what needed to be improved and how can we do it. So what were our methods? We formed a questionnaire and we translated into Arabic, English, and Thai languages. We have about 15% of our dairy workers from Thailand, and we wanted everybody to be able to fulfill the questionnaire. And we also distribute it to all farmers on the farm, and they filled it up voluntarily and anonymously. So we can continue with the slides, Iran. Okay, another one. In total, we had 61 cooperative and 113 family dairy farms selected randomly from all geographical regions. The next stage was the structure of the questionnaire. There were two parts. So the first part was actually asking general demographical questions, such as the responder gender, age, seniority in farming, academic background, past guidance the farmer had on welfare topics, its role on the farm, and the extent of the importance of this topic for him and his enjoyment on work. The second part asked professional questions related to several themes. The first one was the effect of welfare awareness on the productivity. The second one was general knowledge regarding census and social structure of the cattle. The next one was the effect of man-animal interaction on the milk yield. And another important topic was pain perception and prevention. So the way we analyzed the questionnaire, the answers were single choice only. And then we could form an overall welfare assessment score to each respondent in order to cross those answers with other parameters. An additional analysis was performed only on farms that are reporting to the herd book, which gather all the production or productivity data of the specific farm. And then we used a method of gathering a mean welfare assessment score of each work of the farmers on each farm in order to have a score for the farm itself, and then check correlations with other parameters. So what were the results? We had 500 farmers filling up the questionnaires from 174 farms, which account for about a quarter of the dairy farms in Israel. The effect of the demographic variables were quite interesting to see. We first saw that there was a difference in the welfare knowledge between farmers from private family farms and cooperative farms, which could be explained by several reasons, and I will talk about it later. And we also seen that the on-farm self-training for the work led to lower welfare assessment score than training through professional husbandry courses and workshops outside of the farm. So farmers who were trained locally had a mean welfare assessment score of 58, comparing with those who were attending external courses, which scored 66 points. Another interesting thing to see was the higher scores of some duties on the farm. The highest was for the man responsible for the herd health, and after him, the general manager. So they had higher scores comparing to workers who were milking most of their time, or those responsible for the calves. Another interesting thing was to see if the years of experience, the number of years, had some effect on the score. And we see that it has a good effect up to the point of about 30 years of experience. And this was quite surprising to see that farmers work very long duration on the farm tend to have or have significantly have lower scores. It has to do mainly with the fact that many times they cannot not really leave the farm to go to outside courses and guidance, and then their level of knowledge remain quite the same. Then we wanted to see how, of course, the effect of the knowledge of the farmer on the milk yield on the farm. So we looked on the average scores per farm, and we can gladly see that the level of knowledge positively impact the milk yield. We see that the farm with the highest scores are those with the higher average yearly milk yield per cow. So this was very important for us to see and to show to the farmers. And then examining some practical welfare issues on the farm. The first step was to test the knowledge about cow senses. We asked, does the cow hear sounds that we can't? And we saw that majority of the farmers know that yes, it does hear differently than the human ear. The next question regarded to sight. So as we just heard by Aviv, the cows do see things differently, and most of the farmers were quite aware to that, although a quarter of them didn't answer the correct answer, but still majority has some good understanding of its senses. Then when we looked on learning and memory processes, we asked, can a cow recognize a person who treated her in a negative or positive manner on the past? And then we saw that 70% do think the cow can recognize, so it has a long-term memory. The next question was asking, does the cow remember things that happened when she was a young heifer? So here we can see that 57% think it can remember, but some farmers were not sure what was the maximal duration of these memories, and if she can really remember so far. When we asked, can the milker behavior annoy a cow during milking, and we were referring to noisy, annoying, rude behavior, so 94% do acknowledge the fact that it does affect the cows, and this is really due to a good work of the milk board, Israeli milk board, which was training the milkers in the milk parlor during recent years and even more, and it gave some good results, as we can see. When we tested the conception of pain, so this was the million-dollar question, does high milk yield signifies necessarily good animal wealth? So most of the farmers think, yeah, if the cows feels good, she will give a lot of milk, but 36% do acknowledge that it is not necessarily the case due to genetic factors and nutritional factors. Would a cow drop in milk if she experienced pain? So 62% agree with that assumption, and 25% understand that it is not necessarily the case. I can tell you that when we asked veterinarian these questions, they were much more familiar with the complexity of this subject and the idea that it is not always reflected in the milk yield. But when the pain is very visible, like in lameness, for example, does it affect the milk yield? So there is a good understanding that, of course, such a strong acute or chronic pain is very important to recognize and to treat. When we wanted to test the perception regarding mutilation procedures, we asked if cold branding, which is the only kind allowed in dairy farms in Israel, is it a painful procedure? So 63% thought it is painful, and it will be mandatory to use pain medication concerning that procedure in the upcoming regulations. And when we asked, do you think it is important to give a calf painkillers when removing horn buds, which is performed in Israel in the first few days of life by a caustic paste, 72% believe it is necessary. So this goes along very well with the line we see in the regulations to come. The subject of social aspect of calf rearing up to weaning is quite controversial in Israel. And we can see in the next slide that a majority of the farmers are quite conservative and prefer to keep their calves on individual pens up to weaning, mainly to prevent contamination of diseases. But we see also that 38.5% of the farmers do think it is better for the calf to be reared in groups, and 10% think the best way is to do it in pairs. We know, in fact, that 60% of the farmers, when we ask what is the common practice in your farm, so they stated that they do raise them in groups or pairs up to weaning, 60% of the farmers. So to conclude, the farmers demonstrated relatively good awareness to cow senses, to the social structure, which I didn't show you here because we don't have enough time, to cognitive processes. It's fairly well. The domain we saw that need improvement, as we can see, are the topics of pain expression, for example, and how well can farmers recognize when there is a pain in the cattle. And there was also a partial understanding of the complex connection between welfare and productivity. And the last point is that those results that we've seen, which really were showing for the first time on this national large-scale basis, the importance of guidance and knowledge acquisition on the perception of welfare issues in order to, of course, continue and improve those subjects on the farm. So thank you very much for listening, and for Liran for the technical help.
Thank you, Sarah. So there are a lot of questions. The first one is how difficult was it to gain cooperation of farmers to answer your questionnaire?
So actually, it was there was quite good cooperation. We had 3% of the farmers which preferred not to participate on the survey, and some of them were afraid that the student that was coming to do the work on the farm was maybe from Anonymous or some organization like that, and so this was the reason to say no. But overall, beside that, we had a very good cooperation.
And there any plans on implementing what you've learned in some way?
So actually, as I said, the purpose was to point out the subjects which needed improvement. And since then, the guiding extension in the Ministry of Agriculture started a very large project, which is just dealing with that purpose. And they're pointing out the areas for improvement and performing guidance to the guides for the first part, for the first step, and those guides after that are distributing the knowledge to farmers in all branches of agriculture.
I have to say, I know there's a lot of work to do, but I'm cautiously optimistic. I thought the answers would be much worse.
Yeah, actually, we were quite optimistic too, and we have seen that farmers do understand the importance of welfare and its effect on their profits, productivity, or the well-being of the farmer, everything. And they do understand that it's an holistic thing. Everything is connected.
Okay, thank you so much, Sarah. Are there any plans on maybe doing a similar questionnaire with other farm animals, other farmers?
So the extension unit is doing something quite similar for the sheep and goat branch, and I think also for poultry. And it's an ongoing process, so we will see how it continues.
Great. Thank you very much. Chen will just say a few words and then we'll part ways.
Yeah. So thank you. So thank you very much, everyone. And it was a wonderful day and a lot of lectures, and we learned a lot. And I hope to see you next year. And whoever comes tomorrow, we'll see you tomorrow in the third part and looking at the SDGs and looking at the sustainability part concerning under welfare. So see you tomorrow or see you next year. Bye-bye.
Thank you.